Welcome back, everybody. This is part 2 of Chapter 9, Muscles and Muscle Tissue. In the last part, we talked about the anatomy of the skeletal muscle. We also took a look at all the detailed steps involved in getting skeletal muscles to contract, meaning that they were forming their cross ridges and they were doing their power stroke. And as a result of those two mechanisms, they were completing the sliding filament theory. In this part of the chapter, we're going to take a closer look at the motor units that are actually involved and what it means when the muscle starts twitching and how we can have different summations, i.e. recruit more muscle fibers so we can have a stronger overall contraction and relaxation stage. We'll also be taking a look towards the end at smooth muscle, and we'll see what it has in common and obviously how it differs from skeletal muscle. Now, before we kind of dive into it, I just want to introduce you to two terms that you'll see quite a bit as you go through the chapter, and that is isometric versus isotonic. These two terms will indicate how the muscle reacts to its action potential. So what we see happening is, as the action potential comes down from the axon, it will obviously interact with the neuromuscular junction, and it will generate action potentials on the sarcolemma, which will then enter into the individual muscle cells through the T tubules. And as this is happening, the excitement spreads, and as a result, we see that we get an excitability or a response from the muscle, aka the muscle organ all by itself, and that can either be that the muscle will shorten or it will not shorten during its contraction. If you see that there is no shortening, and that is due to the fact that the muscle tension is increasing but it doesn't exceed the amount of load that's being placed on it, we like to call that an isometric contraction. If you see that the muscle does shorten, and this all has to do with the fact that the tension exceeds the load, then we like to call that an isotonic contraction. Let me give you a few examples. So iso, first of all, means the same. Isometric means the same measurement. So the muscle is not going to change in its overall distance, or it's not going to shorten or elongate. So this would be, for instance, whenever you tense up a muscle, the joint will not be doing any movement. A really good example would this be if I were to tell you to go ahead and pause the video and go get yourself a glass of water. So we're going to reach for that glass of water with our hand, and holding the glass in front of us, with our other hand, we're going to start pouring water into the glass. Now, as you're holding the glass, you're obviously utilizing the muscles in your arm to stabilize the glass and preventing it from not falling. But when you look over to the arm, you're going to notice that none of the muscles are actually shortening in their overall shape. So that would be an example of an isometric contraction. Another example would be, for instance, if you guys like the plank when you're exercising, when you assume the plank position, or if you guys do the leans against the wall where you're squatting and your back is against the wall. Whenever you hold that position, that's an example of an isometric contraction. If you do yoga, you tend to do a lot of poses where you have to kind of like extend and hold the muscle. And you'll feel the muscle getting tired because the muscle is contracting, but you don't physically see it moving because it's an isometric contraction. An isotonic contraction is what we're used to seeing kind of like when we do bicep curls. And if you want, if you could start curling up your arms right now, you can notice that your muscle, you can actually feel the belly of the muscle moving back and forth as you pump your arms. And whenever you see that the muscle is physically shortening itself and responding, then you have an isotonic contraction. Of course, the reaction that the muscle is giving is due to the fact that it has a motor nerve that is sending signals down its axon and interacting with the neuromuscular junction. So on this slide, it introduces you to the term motor unit. And a motor unit is going to be the functioning unit between the nerve and the muscle relationship. So this will include the relationship with the neuromuscular junction. And also what we see happening is that it will consist of all of your motor neurons as well as the fibers that it will supply. And a motor unit can supply a few or several hundred muscle fibers. It all depends on the overall size of the muscle as well as the contractile strength that you're trying to generate from that location. So here, for instance, in our illustration, we see we have a cross-section of the spinal cord, and we see that our nerves are radiating outwards. And as you can see, you have your purple and your red line indicating your motor unit number 1 and your motor unit number 2. And as you follow it through, you're going to notice that they make their neuromuscular junctions with the skeletal muscles. And what we see happening is, depending on how many motor units that particular muscle has, it will contribute to the strength that that muscle is able to generate. So for instance, if you only have a handful of motor units dedicated to a muscle, usually that muscle will have a very weak, delicate contraction. So for instance, fluttering your eyelids, or even using your fingers to contract and relax allows for fine motor control of that particular muscle. If you were to increase the motor units, then what you're doing is you're generating more and more strength that muscle is able to have when it contracts. So that would be an example of a more bulky muscle. So maybe the muscles that are included in your hamstring or your gluteus maximus muscle or your gastrocnemius muscle, the muscles that make up your calves. And at the core of it, what we see happening is when you go ahead and you generate that action potential, the simplest contraction that will result is called a twitch. And this is literally one action potential activated by a motor neuron. Twitches are very quick, they will contract quick, and they will relax quick. And usually to the human eye, you're not able to really detect a twitch. And that's because it happens so fast that what we see actually happening are that the twitches start building on top of each other. And what our eye perceives as one smooth muscle contraction and relaxation is actually the result of thousands of twitches just on top of each other. You are able to detect the electrical current of a single twitch using a myogram. And when you do that, you're able to see the electrical current as it's passing through the motor units, and you're able to distinguish how many motor units you are exciting. Are you just doing a few, aka a few twitches, or are you basically maximizing the amount of tension that you can place on the muscle fibers? So here, for instance, we have a typical myogram. And on this myogram, what you want to notice is that you have three particular phases that the muscle is going to go through. You have the latent period, the period of contraction, and the period of relaxation. Now notice how by the time you have a single stimulus that sets off at time 0, and the muscle will go ahead and react to that stimulus. And as it's increasing its tension, what we see happening is that it's going to first encounter the latent period. The latent period is what we were discussing in part 1B, in the fact that once you have excited the muscle on the outside, you now need to do your excitation to contraction coupling. And as you're doing that, the action potential is spreading across the sarcolemma, entering into the muscle cell through the T-tubules. While this is all happening, you don't have any muscle tension that you're observing because you haven't formed any cross bridges yet. You're still in the stage of exciting the actual muscle. By the time the latent period is over with, you have formed your cross bridges and you start doing your power stroke, which means that your muscle will actually start generating enough tension that you can see it moving, and this will then be your period of contraction. Obviously, after contraction, we'll have a period of relaxation, and that is usually not only indicated by the fact that you lose your cross bridges and your power stroke, but also due to the fact that the calcium is removed and it re-enters the sarcoplastic reticulum. And as we all know, when we remove the calcium, then the troponin glides to tropomyosin back over the actin site, thereby preventing the myosin head from binding to the actin sites. Here's a comparison of the three stages if we were to compare our muscle. Now what they're doing is they're going to start comparing a very small muscle like our extraocular muscle, which is usually found around the eyeballs. Excuse me a second. This tends to have a very rapid and brief response. Then we're going to compare it to the gastrocnemius and the soleus. Both of these muscles are found in the back of the leg, lower leg, and what we see happening is these muscles, because they are bulkier, they will have a slower response, but they will be able to hold on to their contraction a little bit longer than the fine, delicate muscles. We also see that we have a graded muscle response. And this is all due to the fact, as what I was mentioning to you before, that at the core, when you activate a muscle, what you're generating is a twitch. But we don't see the twitches, and that's because usually the stimulus that initially sets off the twitch will be amplified so that the twitch starts to increase and your response becomes more dramatic. It becomes more amplified. And the way we can do this and generate a graded muscle response so that the entire muscle as an organ contracts or responds is that we can either change the frequency of the stimulation or we can change the strength of the stimulation that the muscle is being exposed to. So let's take a look at this little diagram right here. And what we're doing here is we're comparing the stimulus into the tension that it's able to generate. So you have your little red arrow at the bottom left-hand corner of the graph, and you can see that the stimulus is enough to get the muscle fibers to react. And what the muscle fibers will do is they will have a contraction. They will form their cross bridges, they will do their power stroke, they will do their sliding filament theory, and after that, when the calcium is removed, the muscle will go ahead and relax. And what you're looking at here is a single stimulus leading to a single twitch. But as I mentioned several times before, we don't see that. We see more of an amplified result. So how can we increase our twitches so that we can see the muscle contract as a whole? That's where our first option comes in. So over here on this option, we're looking at increasing the frequency of our stimulation. Now this is going to be a low stimulation and frequency, meaning that you're going to have a little bit of relaxation time between the stimuli. So go ahead and look at your graph and identify the 4 red arrows. Each of them will represent the stimulus. Now the stimulus will stay the same in terms of the voltage. What you're going to do is you're going to increase how many stimuli the muscle is exposed to. So compared to our last graph, this graph will have 4 exposures to the stimuli. And as you can see, the arrows are spaced away from each other. And the space in between will indicate a time of rest where no stimulus is received. Now, if you now look at the blue line that is generating and showing us the tension in the muscle, what we're going to see is when the initial stimulus sets off, the muscle will start to contract. And as it starts to go down to relax, it will receive the secondary stimuli before it actually reaches its resting membrane potential. So it will only be able to do a partial relaxation before it's exposed to a second stimuli. So the second stimuli actually causes an increase of calcium to be flooded into the cell, and thereby the muscle is able to react even more aggressively. And what we see happening is that the contraction strength or the contraction tension increases more than it did the first time. And we see that same pathway follow, that the muscle will partially start to relax after the second stimuli. The third stimuli comes in and it generates even more tension for that muscle. So you have what we like to call a wave or a temporal summation that you're seeing. It almost looks like a little stairway where your steps are increasing, and that's basically meant to represent the fact that you're doing a wave summation and you're basically recruiting more and more muscle fibers, and this is going to lead us to produce a very smooth, continuous contraction, which is what we tend to see with the naked eye. So your smooth contraction that you're witnessing when you're doing your little bicep curls is actually the result of these partial relaxation steps by increasing the frequency of the stimulation in a low frequency pattern. This will result in also what we like to call unfused or incomplete tetanus, meaning that you activate the muscle fibers in different fractions up until the point where the muscle is fatigued and it will go ahead and relax as a unit. And that is what's shown with the line pointing downwards between the 200 and 300 millisecond time periods. Now, what if we take the same scenario of exposing the muscles to a stimuli, but instead of doing it a low stimulation frequency, how about we do a high stimulation frequency? So we're going to go ahead and move those red arrows closer together, and we won't allow the muscle enough time to relax or rest in between stimuli. So here's that little scenario. So this is an example of a high stimulation frequency. So go ahead and find your red arrows. Notice that once again, the strength of the stimulus is the same. What makes it different is now we're going to increase the frequency, but we're increasing it in such a dramatic pattern that the muscle has absolutely no time to even partially relax. So what we see happening is because there is no relaxation in between the stimuli, the muscle will go ahead and do what they call complete tetanus, which is that all muscle fibers will go ahead and interlock, they will contract, and the tension will plateau up until the point that muscle fatigue is reached, and then the muscle will go ahead and relax as a complete unit. The other way that we can increase the stimulus strength is obviously also by increasing the voltage that the stimulus does. So not the frequency, but the voltage. And that's what this diagram is going to show us. So notice how you have three graphs that you'll be comparing. The top part will take a look at the stimulus as we're increasing the voltage. So notice how you have your red arrows once again, and the red arrows keep getting longer and longer as you go from 1 to 10, indicating that we're going to increase our voltage or the amount of stimulus that we're exposing the motor units to. The second diagram shows us a cross-section of motor units, and notice how you have your little filaments indicated in the middle, and what you're looking for is how much of these units am I actually exciting as I'm increasing my voltage. And at the bottom graph, it's going to show you the actual muscle contraction. So the blue line will indicate the tension that's being shown by the muscle, and you'll be able to track how much of a contraction is actually seen. Now, what we're going to alter here, keep in mind, is we're going to alter the voltage of the stimulus. So let's go over to the left-hand side, and let's take a look at motor unit summation. And what we see happening here is that as we start to manipulate the stimulus voltage, we're going to have some that fall at sub-threshold stimulus, meaning that the voltage is too low to recruit over any motor units, which means that you won't have any contraction at all. Some will have a threshold stimulus, meaning that this is the minimum amount of stimulus, minimum amount of voltage that's required to get the first noticeable contraction. And then obviously there will be a maximal stimulation. Maximal stimulation indicating the fact that you're at a high enough voltage that you have recruited all of the contractile units, all of the motor units, so the muscle simply cannot contract anymore. It will not give an additional graded response. It will just keep stimulating its contraction to its maximum capacity. Let's go ahead and look at the graph, and let's see if we can find our subthreshold, our threshold, and our maximal stimulus. So go ahead and start with number 1, and what you're going to do is you're going to look at the top graph, and you're going to notice that they have a little white dotted line for you that's been labeled the threshold stimulus. All right, now the threshold stimulus, as it's indicated in writing on the left-hand side, that's going to be the minimum amount of voltage that's needed for us to get a noticeable contraction. So as you can see, number 3 is reaching the threshold. Anything that's below 3, so 1 or 2, that's going to be a sub-threshold. It falls below the threshold, which means that even though I'm stimulating it with the nerve, I'm not recruiting over any of my motor units, so my muscle is not contracting. So go ahead and look below the 1 and 2 in the second illustration, the proportion of motor units excited, and you're going to notice that all the little filaments are the same color. No portion of the motor unit is changing into that darker pink color, which means the stimulus is below threshold. And that also means that if I look at my bottom graph, I'm going to notice a flat line because I'm not contracting any of my muscles. I'm going to see a little blimp of contraction in my third voltage or stimuli because if I look at my motor unit cross-section, I see that I've recruited some of the fibers, and as I'm recruiting the fibers, 2 of them to be exact, I see a little twitch that starts to generate. And from there on, as I start increasing my voltage for the stimuli, so I'm moving on to step 4, 5, 6, and 7, I see that in my cross section, I'm recruiting more and more of the motor unit. Notice how more of it is starting to change into that darker color. And the more motor units you recruit, the stronger the muscle contraction becomes. So you can see that stairway pattern starting to develop in the bottom graph, where the contraction is getting stronger and stronger as you increase the voltage strength. Now, eventually you're going to notice that you're going to kind of plateau. It's not going to increase anymore. And that's going to correspond to the fact that number 7 indicates our maximal stimulus. This is where our entire motor units are excited. They're fully saturated. So that means that it makes no sense for me to continue on with 8, 9, or 10, because even though I'm increasing the voltage, there are no additional muscle fibers to recruit, which means that my contraction strength will not be altered when I compare it to what is happening with my 7th stimuli. The other thing we should kind of discuss is the fact that part of our homeostasis is to maintain muscle tone. And this means that all your muscles are consistently slightly contracted. And the amount of contraction will all depend on body build. It will also depend on which muscle groups we're looking at. So for instance, around our abdominal area, next week we're going to talk about the fact that you have your external obliques, your internal obliques, your transverse abdominis that kind of layer on top of each other, creating almost like a girdle that wraps around the abdominal cavity. These muscles are consistently being contracted because they form a protective layer that protects the digestive organs that are inside of it. The consistency of this contraction is often controlled by our spinal reflexes, and the goal of it is to basically maintain our overall body built, as well as maintenance and protection of the organs that it is trying to protect. It will also obviously play a role with our posture and allowing us mobility once stimulation is received. So here's a mention. I think I should have probably moved this picture closer to the start of our lecture. This picture shows us the isotonic versus the isometric contraction that we started this part of the chapter off discussing. Remember that isotonic means that you will change the length of the muscle. So as you can see in the picture, the muscle will start to shorten, quote unquote, and that is because it's actually moving the belly of the muscle. Whereas a isometric contraction means that the muscle length stays the same, and usually that means that the load is greater than the maximum tension. So the muscle is still contracted, but it will not actually shorten or lengthen. And in fact, what you can do is you can take a look at the graphs that are at the bottom, and the muscle length, which is the bottom graph, you can see the little green line. Notice how it alters or the shape changes when you're doing isotonic, but when you're doing an isometric, because the muscle length stays the same, the line kind of flat lines from beginning to end. All right, so when it comes to factors of muscle contraction, and we take a look at the actual force of the contraction, we know that it obviously has to do with the amount of cross ridges that you'll be able to produce on the microscopic scale. So thus far, we have kind of discussed a few of the factors that can affect it. Obviously, the first one that is here on your PowerPoint, it says the amount of muscle fibers that you're able to stimulate. And that's what we were talking about beforehand with your recruitment effort. The more motor units you recruit, the greater the force. Another thing that you might want to consider is actually the size of the fibers. How many muscle fibers are actually found within the organ? And the bulkier the muscle, the more tension it can develop, which usually allows for greater strength movements. This is often what we see with people who are who like to bodybuild or weight lifts. We see that the muscles will start increasing in size. They'll do hypertrophy, and as they enlarge, they become bulkier. They have more muscle fibers, which means they're able to generate more tension and more contraction force. We also discussed the frequency of stimulation. So the greater the frequency, the greater the force. And this was when we talked about the fact that you can have a summation that will either lead you to your incomplete, or your complete tetanus. It all has to do with the spacing between the stimulation frequencies. And then the last thing that will control the amount of cross bridges being formed is actually something that's very important to our homeostasis, which is the degree of muscle stretch. The degree of muscle stretch will take a look at how much difference there is in sarcomere length as the muscle starts to contract and relax. And this all will have to do with how easy it is for the initial cross bridges to form between the thick and the thin filament, and how much space they will have to go ahead and do their power stroking, aka how much space do they have to control the distance between the two Z discs that make up the sarcomere. I think we can probably look at this illustration to try to explain the length-tension relationship between the sarcomere and the muscle contraction force. So what you're looking at over here is a diagram that's showing us three different scenarios, and it's showing you your initial sarcomere length, and it also shows you the amount of overlap between the thin filament in blue and the thick filament in that orange shade. So let's go ahead and let's start off with the middle one. So it says sarcomere at resting length, and it says when the sarcomere is at 100%. And what that means is that it's basically at its normal length. And at the normal length, we have optimal performance of generating a very strong contraction force because your thick and the thin filaments are overlapped just enough that they can easily form that cross bridge, that physical connection. But look how much space there is on that thin filament before you get to the Z disc, that little black line. Because of that space, that will allow you to generate lots of power strokes, which means that the muscle can fully contract and eventually can fully relax back. Our other option is if you have a sarcomere that's already shortened, meaning already compressed even before it gets stimulated. So in that case, we're going to look on our left-hand side to the area that's indicated by 75%. And what we see happening here is that the sarcomere is compressed, it's shorter than it normally is. The thin and the thick filaments are fully overlapping with each other, which means you won't have a problem making your cross bridge, but there is no free space on the thin filaments for your power stroking to occur, which means that the muscle will not be able to generate a stronger contraction. This would be sort of me asking you to do a bicep curl and then telling you as your muscle is fully contracted to go ahead and increase the contraction force of the muscle. Once all the muscle fibers are entertained and all the cross bridges are formed and there's no more room for the power stroke, you can't generate more of a tension within that muscle. The other extreme of that would be located towards the right-hand side when we see that the sarcomere is excessively stretched at rest, which then means, and this is the one indicated by 170%, so it's a lot longer than it normally will be. In this scenario, what we see happening is that there is barely any overlap between the thick and the thin filament, which means that they will have a hard time or a difficult time making that initial cross bridge. And because it's so difficult to make that initial cross bridge, even though they have plenty of space on the thin filaments for the power stroking and the sliding filament to occur, the muscle will have to put in a lot of effort to go ahead and generate enough tension for it to do the power strokes, all because it's already excessively stretched and it will be very difficult to make that initial physical contact. The best way to think about this is if you've ever heard the expression that you should bend through your knees, not your back. So in your back, you have this really long muscle called the erector spinae muscle, which you'll see in a few weeks when we kind of look at naming all the different muscles in the body. And what ends up happening is, let's say you have a heavy box that's sitting in front of you. When you bend through your back, we see that the erector spinae starts stretching. So it starts to look like that 170% on our diagram. This erector spinae can be overly stretched, which means that by the time that you get to the box, if the box is really heavy, you can't generate enough contraction strength in that erector spinae because your thick and your thin filaments are having a hard time making their cross bridges. Because of this, you'll feel a sharp pain in your back. On the other hand, if you bend through your knees, then what we see happening is that you're utilizing the muscles in your hamstring and in your gastrocnemius. And by the time you do that, you're not only recruiting a much bulkier muscle, by the time you get to the box, you still have lots of muscle fibers that you can interact with that haven't been activated and that sarcomeres are still in a resting length. Anywhere between 80 to 20% is perfect. Thereby it's able to give you a nice, smooth, strong contraction that you won't notice the next day that your body aches from because the muscle was never overly stretched. Alrighty, so our last few slides are going to take a look at smooth muscle. So previously we talked about the fact that smooth muscles can be found in the viscera, so the internal organs, and we also discussed the fact that it's under involuntary control and it is a non-striated muscle, and that's because they will not have a very uniform pattern between the thin and the thick filaments. We also see that this muscle will only tend to have one nuclei per individual cell, where the skeletal muscles is multinucleated due to the fact, obviously, because of all the strength that it has to generate within the contractions. Oh, I should also mention, as it says at the bottom of the PowerPoint, that when we look at the smooth muscle, it will not have all the different connective tissue sheets that we saw in skeletal muscle. Instead, it will only have the endomyosin. Your smooth muscle will also tend to lay in two different patterns. It will lay in a longitudinal layer where the fibers run parallel, contributing to the length of the organ, allowing for movement from point A to point B. So for instance, if you imagine your GI tract, your digestive tract, imagine your your short, your small and your large intestines. My apologies, completely forgot the name of those organs. If you imagine your small and your large intestines, those are nothing but very long coils, and the length of it is composed of the longitudinal layer of the smooth muscle. The smooth muscle can also be used in a circular layer, and this is often then used to create a lumen or an opening within the organ that we can easily control its constriction and relaxation stage. So this will come into play, for instance, as the food is moving through your intestines. We can make the lumen smaller or larger, controlling how much mixture of digestive enzyme the food comes in contact with. When it comes to smooth muscle and the digestive system, you will also see the term peristalsis quite a bit. Peristaltic movement is an uncontrolled smooth muscle movement that will create almost like these wave-like patterns that will be utilized by your digestive system to move food all the way from your esophagus, through the stomach, down through the small and the large intestines, allowing the food to be mixed with the digestive enzymes and obviously for digestion and absorption to occur within the GI tract. So here in this illustration, if you go ahead and take a look at B by the cross section, you can see the longitudinal format and you can see the circular format of your smooth muscles layering. So they can go lengthwise, which will allow movement from A to B, and they can go in a circular pattern, which will allow us to control the lumen or the inside of these hollow organs. I should also mention that when it comes to smooth muscle, it does not rely solely on nerve innervation, which we saw in skeletal muscle, so it will not have a neuromuscular junction. Instead, we see that smooth muscle can be activated by anything from a hormone to a neurotransmitter to some local chemicals. And yes, they can also be activated by nerve innervation, but that is not their only option. In fact, what we see happening is that there's lots of examples where the nerve fibers will interact with the smooth muscle, but since they don't have a neuromuscular junction, they're going to use these areas called varicosities, which will kind of look like little bulbs that are swollen all along the length of the muscle fibers. Let's take a look at this on the illustration. So here we go. So here we have our smooth muscle on top, and all those yellow bulbs that you're seeing, those are the varicosities. And when the signal comes down the axon, it's going to cause neurotransmitter to be released, and it's going to be a much wider cleft than you would see in the neuromuscular junction when you compare it to your skeletal muscle. So oftentimes they'll talk about the fact that the transmitter will diffuse into the opening and make contact with the smooth muscle. So they like to call it a diffuse junction. So varicosities and diffuse junctions, whenever you see that terminology, please keep in mind that describes the relationship between the nerves and smooth muscles. We also see that your smooth muscle will not have sarcomeres, it will not have myofibrils, and it will also not have T-tubules. It will have SR regions, sarcoplastic reticulum regions. These will still function in calcium release. However, what we do see happening is that the SR region will store some of the intracellular calcium, but the majority of the calcium will come from an extracellular component when the smooth muscle needs to contract. That is a huge difference when we compare it to skeletal muscle, which solely or mostly relies on intracellular calcium release from the SR region. In fact, the sarcolemma in your smooth muscle has these areas called calveola, which are these little infoldings, and they are filled with calcium channels that will allow the extracellular calcium to come flooding inside the cell at a high rate of speed so that the smooth muscle can go ahead and cause its contractions. Another difference that we see between the smooth muscle and the skeletal muscle is the composition of its thick and the thin filaments. Now, both of these muscles will have the myosin filament and the actin filament, but what we see happening is that usually when you look at the abundance of thick to thin, in skeletal muscle you would have a 1:2 ratio, meaning for every one thick filament, you would have 2 thin filaments. In smooth muscle, that ratio is 1:13. So the thin filaments far outweigh the thick filaments when we look at smooth muscle. They will also not be arranged in a very uniform pattern. They will crisscross and be very abstract in their overall layout within the smooth muscle. We also see that they do not utilize troponin. They do have tropomyosin, but they don't have the troponin on top. Instead, they're going to utilize calmodulin to regulate the calcium binding. So troponin is not found anywhere in smooth muscle, but tropomyosin is found in both smooth muscle and skeletal muscle, and calmodulin will come in for the smooth muscle since it does not have troponin. Now, since your thick and your thin filaments are arranged in such an abstract pattern, what we see happening is when the muscle contracts, it doesn't contract in a nice linear pathway. Instead, you're going to have almost like a little corkscrew mechanism. So if you take a look at the illustration all the way at the bottom, what it's showing you is a little muscle cell that's contracting. Notice how the arrows are coming from either side, and that's just showing you how it can kind of have the corkscrew mannerism. And the muscle cell will actually kind of look like it puffs up as it contracts and as it relaxes. So it's a very kind of like uncontrolled pattern in its reaction to the muscle contraction. We also see that it will have some what they call dense bodies. You can see it labeled on the illustrations. Those are going to be little proteins that anchor the filaments to the sarcolemma. So they'll have more of some regulatory proteins that will anchor the contractile unit of the muscle. Now, when it comes to the actual mechanism of contraction, smooth muscle will tend to have more of a slow, synchronized contractions. Their cells will use gap junctions to allow for them to be electrically coupled. We've discussed the fact that the cells do not have to rely on nerve stimulation. Some of the cells, in fact, are what we like to call pacemaker cells, meaning that they're able to initiate their own depolarization and they're self-excitatory, so they don't need an external stimuli to start the action potential process. And we also see that if we have some similarities between skeletal and smooth muscle. So go ahead and take a look at the bottom of the PowerPoint, and let's take a look at what they have in common. So 4 points to highlight. The first one is that you still have a sliding filament mechanism, and you still have a thin and a thick filament. And it's still made out of actin for the thin, myosin for the thick. The second point is that you still need calcium levels on the inside of the cell to be high for you to have your initiation of your cross bridges. You will still need ATP to maintain your contraction. So that's the third thing that they have in common. And the fourth thing is that when the calcium drips down or the calcium stops being secreted to the inside of the cell, we see that in both smooth and skeletal muscle, the contraction will come to a halt. And that is because the tropomyosin will cover the actin binding site, preventing the cross bridges from forming. So these are the four items that the skeletal muscle and the smooth muscle have in common. Let's take a look at some differences. And we've already mentioned a few in the previous slide, but here are some other ones to kind of think about. So we already discussed the fact that some calcium will still be stored in the sarcoplastic reticulum in smooth muscle. However, the majority of the calcium will actually come from an extracellular location. That's a huge difference because, as I mentioned to you before, skeletal muscle will get the majority of its calcium from the intracellular storage of the sarcoplastic reticulum. I've also mentioned to you that calcium will bind to calmodulin, not troponin, and that's because troponin is not found in smooth muscle. I haven't really touched upon the third item, which is the fact that when the calmodulin notices the calcium, it does have a slightly different way of activating the myosin kinases, which will cause the reaction of the cross bridges to start. And we also see that the myosin kinases will play a role in phosphorylation, meaning utilizing the ATP molecules to cause the shifting of the myosin head. So the mechanism of forming the cross bridges is slightly different in your smooth muscle than it is in your skeletal muscle. That's what the third and the fourth item highlight on our list of how the smooth muscle is different from the skeletal muscle. Lastly, at the bottom of the slide, it does discuss the fact that if you want to halt a contraction in smooth muscle, it usually requires a few additional steps than in skeletal muscle. For instance, we see that the calcium does have to detach from the calmodulin. We also see that we have to rely on active transport for the calcium to be pumped back into the sarcoplastic reticulum and also out of the cell. So that's where the extracellular will come in. And we also see that we need to have dephosphorylation of the myosin. It is only then that the myosin head will become inactive and it will go ahead and completely release itself from the binding site of the thin filaments. All right, now having said that, that is going to conclude our Chapter 9 discussions. I know I've said it many times before, but please take your time as you go through the video, make your notations, and if you have any questions, comments, and concerns, I would love for you to share them with me through our discussion sessions. But obviously, you can also feel free to email me if you don't want to have the rest of the class see what you're asking. Either way, it works just fine for me. All right, guys, stay safe out there, and we'll talk soon.